Okay, chapter 2, altered cellular and tissue biology, environmental agents. So cellular adaptation. So cells adapt to protect from injury. Central to most disease states is cellular adaptation. These cellular changes can present as atrophy, hypertrophy, hyperplasia, atypical hyperplasia, and metaplasia. These are discussed in greater detail in your chapter, and I'm going to refer you to that section. So cellular energy. Cells become injured when homeostasis is disrupted, and there are a plethora of reasons this can occur. Once cells are injured, this can result in tissue and organ injury and possibly disease. Now, this can be reversible or irreversible. I'm going to refer you to table 2.1, which goes over progressive types of cell injury and responses, and you can kind of read that on your own. Cell injury or death occurs when there is a disruption in the cell membrane, lysosome damage, mitochondrial damage, lack of protein synthesis, and enzyme leakage. So if you think about this, if you have these kind of damages, these cells can't function. So if you had mitochondrial damage, you can't produce ATP, which is necessary for many functions in the body. For example, we need ATP for skeletal contraction to be able to move. So without it, those kind of characteristics of life would not occur. So I'm going to refer you to table 2.2 and figure 2.7 and 2.8 for the next few slides that I'm going to cover. Ischemic and hypoxic injury. Hypoxic injury is the most common, and the common cause of hypoxia is ischemia. And then inflammation is induced by the hypoxia. Ischemic injury is due to arterial narrowing and eventually blockage by blood clots. A lack of oxygen halts the aerobic process, which results in less ATP availability. So without oxygen, the byproduct of the breakdown of glucose, pyruvic acid, cannot be brought into the mitochondria. So therefore, if that can't be brought into the mitochondria, then you can't produce ATP. So you're in an anaerobic process with a lack of ATP, and there's a buildup of lactic acid. So the breakdown of glycogen yields ATP, but eventually glycogen is depleted and the anaerobic process stops. A lack of ATP results in a failure of the sodium, potassium, and calcium cell membrane pump, which then results in accumulation of sodium and calcium intracellularly, and the cell swells. Potassium then moves to the extracellular space. There is swelling of the rough endoplasmic reticulum, so therefore ribosomes detach. And if ribosomes detach, you can't have protein synthesis. Without protein synthesis, cells cannot repair. If oxygen is restored, all of this is reversible. However, if oxygen is not restored, there is vacuolation, lysosomal swelling, and mitochondrial swelling. The presence of calcium in the cell causes various enzymes to leak, which disrupts your cytoskeleton and cell membrane, promotes inflammation, DNA breakdown, and ATP depletion. So without the proper organelles, enzymes, cell membrane integrity, and ATP, the cells cannot perform their daily tasks, and you lose that homeostasis, and then cells can die. This also, again, is outlined in figure 2.7 and figure 2.8 if you need a diagram to show you what I just went over in the slide. So now, ischemia reperfusion injury. So there are diagrams to accompany this. You can look at figure 2.13, figure 2.14 and figure 2.15. So we have xanthine dehydrogenase is converted to xanthine oxidase during reperfusion with oxygen. The excessive ATP from the ischemic process accumulates purine catabolites, hypoxanthine and xanthine. These catabolites are converted to large amounts of superoxide and hydrogen peroxide by xanthine oxidase. These free radicals can further accumulate calcium in the mitochondria and cause cell membrane damage. There's opening of the large pores in the mitochondria, and there's a release of a large amount of ATP. And this occurs from sudden pH changes, accumulation of calcium, the inflammatory process, and a number of other cellular changes, which then ultimately results in apoptosis, which is cellular death. Free radicals and the reactive oxygen species. So a free radical, so this is a single unpaired electron in the outer orbit. This makes a molecule unstable and it will try to attach to another molecule to stabilize. It can attach to DNA, RNA, proteins, lipids, carbohydrates, and alter the integrity of these molecules. Oxidative stress, this means there's not a balance between the reactive oxygen species and antioxidant defenses. If you look at table 2.3, this shows you the biologically relevant free radicals. And then table 2.4 goes over some antioxidants to counteract this process. So if we look at figure 2.13, this is showing you where that reactive oxygen species is coming in, causing DNA damage and mutations, and then ultimately leading to the pathology that you see clinically. Then in figure 2.14, you can see where the antioxidants, the SOD, which is superoxide dismutase, and then you can see catalase and the glutathione peroxidase, they are changing these free radicals into something that is not harmful. So you can see the SOD changing the hydrogen peroxide, and then you get the catalase coming in, and you ultimately just get water. So it's not harmful to the cell. Then if you look at figure 2.15, again, you can see these free radicals coming in, the superoxide, hydrogen peroxide, and hydroxyl radicals. If they don't get converted, they can lead to lipid peroxidation, protein modifications, and DNA damage, ultimately leading to membrane damage, breakdown, misfolding, and mutations. But if you have antioxidants coming in, such as the superoxide dismutase, you can convert these to harmless products, and therefore you remove those free radicals and you spare the cell from becoming damaged. That is it for chapter 2.